Pixin is bringing nature's 4.5 billion-year-old lighting solution into modern screens. But why? Let's take a trip back in time. These blobby critters on the left are cyanobacteria, single-celled organisms that have been cruising up and down the water column for about 3.5 billion years. And this handsome fellow on the right is a mantis shrimp, and I encourage you to look up more about him. But my question to you is, what do ancient cyanobacteria, mantis shrimp, and humans have in common? All use an opsin and a retinoid pigment molecule that change polarization state in response to light to survive, regulate health via metabolic processes, and keep time. The opsin paired with an absorbent photopigment protein has an activation energy curve. The energy required to activate is tied to a wavelength of maximum isomerization potential. And so for billions of years, life has been using light to do stuff without eyes, like split water molecules, extract stored energy, captured electrons that are then stored in a molecule called ATP, and burp out a waste product called oxygen. Very resourceful cyanobacteria and bacteria can still utilize huge swaths of the spectrum, spanning from short UV all the way to the near infrared wavelengths to this day. From the beginning up until the later advent of primitive animal life forms around 700 million years ago, the mechanisms underlying activation are fairly conserved. For those earliest single-celled creatures, light and dark was the best resolution available. But as evolution proceeded, tinkering with the molecular machinery eventually gave rise to dim light detection and then color vision. Information via photons is used to switch between daytime and nighttime activities. Gene activation is orchestrated dawn to dusk via light cues. Regulation of on and off switches is controlled by structures that are much like the earliest receptors, which in turn led to diurnal regulation of energy metabolism, cellular division, and chromosome architecture modification. Animals arise about 600 million years ago, along with intrinsically photoreceptive retinal ganglion cells, IPRGCs, to carry on adding gene regulation and diurnal activity orchestration. Then come along rods for dim light and cones for color vision, and then some intense diversification during the Cambrian explosion. Pinhole type led to mirror type, led to camera type eyes. Precision around imaging, as well as tinkering around the wavelengths most conducive to survival, can change throughout continued chasing of fitness. But the machinery underlying sight remains primordial. Compared to many animal life forms, the visual perception of humans, the primate that became dominant on the scene about 300,000 years ago, is quite narrow. Only 3 cones compared to the 16 in mantis shrimp. The restricted consciousness allows humans to thrive by screening out distractions, focusing only on clear and present danger or the most valuable resources. Concurrently, fire was then used as the first cultural tool of light and as black body radiating. Then, about 150 years ago, the innovation of incandescent light lamps moves humans to a state of dusk, where lux indoors is generally orders of magnitude weaker than outdoors. This 1884 book cover shows examples of cutting-edge lamp bulbs from Thomas Edison and 3 other inventors of the age, all striving to make the narrative of life less naked and afraid and more Downton Abbey. Since displays come on the scene, the light our eyes experience can be manipulated in all kinds of mind-boggling ways, like this 580,000 square foot LED screen displaying up to 256 million colors at the Sphere in Las Vegas, with the express goal to distract, delight, and disrupt. With these amazing innovations and technological advances, our screens now display the most amazing images and can transmit the most incredible information and keep us connected. But we don't want that to be at the cost of our health. We don't need our screens to make us sick, tired, and stupid.